This video is going to be much shorter than the aerobic respiration video because we've already learned a lot of the preliminary steps, but we'll briefly introduce you to anaerobic respiration. So hopefully you remember that anaerobic respiration, let me just grab a thing to write on here, 1 second. Anaerobic respiration, still being a respiration, right, requires an electron transport chain, which has to be in the membrane. Anaerobic respiration uses an external electron acceptor, so not a compound that is used by the cell for something else. But this electron acceptor can very definitely never be oxygen because we are doing anaerobic respiration. So that is anaerobic respiration in a nutshell. Bacteria are all over the place, right? And so we are going to just assume that the only difference between anaerobic and aerobic respiration is the final electron acceptor. In reality, there are bacteria with all kinds of differences, but it's too much to learn those, so we're just not going to. So we will assume that the bacterium is using EMP glycolysis. We are assuming that the bacterium is using pyruvate oxidation, which I just kind of lumped in with the Krebs cycle on this slide. And then that the bacterium is doing an electron transport chain. So, oh, I forgot to introduce you to this word before. Okay, so chemiosmosis is the protons in, right? And so if ATP is produced by chemiosmosis, then that is the process, well, actually it's the ATP out and in, or the hydrogens out and in. Chemiosmosis is the process of moving protons out to generate the PMF and then moving those protons back in to do work. And it is in conjunction with oxphos normally. So again, our biggest difference in anaerobic respiration is that the electron acceptor is not oxygen, but we're still doing electron transport. So this gives you, you can look at this more closely on the actual PowerPoint on Moodle, but this just gives you an idea of possible electron acceptors besides oxygen. This is just if you care, like I would tell you what the electron acceptor was if you needed to know. All right, so this is going to need to be zoomed in. A slightly different version of the reduction potential chart. Okay, so here is glucose up here, here is oxygen down here. Here is a sulfur thing, here is another sulfur thing. Here is an iron thing, here is nitrate, here is another nitrate thing. And so the important bit here is you guys will remember, even though the electrons are all starting up here at glucose, the distance that the electron moves is directly connected to how much energy is made available in the PMF to produce ATP. So the farther the delta E naught prime, remember delta is change, so the change in values, aka the distance that it moves. So the larger the delta E naught prime, the more ATP will be produced, and the faster the cells can grow. And so the faster the cells can grow in a finite period of time, the more cells you'll have, right? So if the cell can use oxygen, you'll always have the most cells and the fastest growth rate with oxygen. And then for these anaerobic respiration options, you'll have a slower growth rate and fewer cells. So on the exam, I would give you like a reduction potential chart. It would be like that part, right? So it would have the compounds and the values, and then it would just ask you, like, which one do you think would grow the fastest, and how can you explain this? And so I feel really confident that you guys will be able to do that. All right, so our summary for this. Does anaerobic respiration use oxygen? No, it does not. What is happening to the carbons? So if we consider the entirety of the process from EMP glycolysis all the way through the end, then we are going to start with the 6-carbon glucose and go all the way to the 6,1-carbon CO2. And so this whole process will give us a complete oxidation. because there are no bonds remaining between the carbons for us to break. The initial electron acceptor is, the initial electron donor is glucose. The final electron acceptor is going to be something external, not oxygen. Almost always, this is inorganic. Very unfortunately, not actually always. Almost always, but not always, which is why I am forced to say external and not inorganic. The electron carriers are the same. They are NADH and FADH2, and then we will have prosthetic groups in the electron transport chain. So again, considering the entirety of the process from glycolysis on, ATP is made a little bit in EMP glycolysis, and in the Krebs cycle is made through substrate level phosphorylation. Quite a bit is still made at the end of electron transport through oxphos. Do remember that it is called oxidative phosphorylation because the initial energy to create the ATP came from oxidizing bonds, not because oxygen is involved. So we still use oxphos in anaerobic respiration. So how much ATP would be made? Always less, because oxygen has the most, I have to tell you guys, I always get this backwards and have to look, most positive E naught prime. And so you could explain electrons would move the biggest distance. And if you were working with the chart, then you could like draw some arrow to explain this, right? Okay, that is anaerobic respiration.